I'm representing a large team of people. Two of them are here. We have Delia Grace, who has been our main supervisor, and Sally Bukachi, who has been doing work on anthropology. So we have other team members from the Kenya Medical Research Institute, which was doing entomology and some human work. Also people from the Department of Veterinary Services who are in charge of the veterinary aspects of the work. We also had an ecologist from University of Nairobi. Our work was to look at Rift Valley fever in an irrigation setup. So I'm sure maybe most of you know the disease already. It's a it's a mosquito-borne infection, which affects mainly livestock with occasional spillovers to humans. As we know it, it's associated with rainfall. We usually have El Nino rains, which causes flooding, and that flooding allows mosquitoes to come up. But the questions which we had under this project was, if you introduce irrigation, especially in those areas with high risk for the disease, mainly in dry areas, are you likely to provide conditions that are conducive for endemic transmission of the disease? So that you are changing from what we are used to seeing in terms of heavy precipitation to a situation of irrigation where you have endemic transmission from time to time. We superimposed that hypothesis on the issue of ecosystem services. So we are thinking, in an irrigation setup, you are introducing water and food to populations which are challenged because they are now relying on irrigation instead of rain-fed agriculture. But in doing so, there are trade-offs in services in terms of introduction of disease and also in terms of exposure to agrochemicals, which we'll be seeing in a short while. So the picture there gives you the study site which we used, where you have canals bringing water, and of course farms where they grow maize, rice, and cotton, and other things. So for the methods, we focused mainly in the arid and semi-arid region in Kenya, that's northeastern Kenya, which is known to be high risk for Rift Valley fever. We used two main irrigation schemes, which one is called Bura, the other one is Hola, but we soon realized that there are many other small-scale irrigation schemes in the area, which when you take their sum total of the effects on the environment, then it might actually be much more than just looking at the commercial irrigation schemes. So we did entomological studies with an ecologist. We also did participatory surveys and socioeconomic surveys to really understand people's perspectives in terms of what the challenges and also the exposures which they have within the irrigation scheme. We did sero-epidemiological surveys, sampling livestock, that's cattle, sheep, goats, and also humans. We got ethical approval from the African Medical Research Foundation. After all those, when we had all the findings together, we convened meetings with decision makers to share the findings and sort of like help them to integrate these findings into their routine disease control policies. So that's the map for the study site and showing the irrigation scheme in deep green. So for the first analysis, that's on land use, land cover changes. We compared the current, not really current, but 2010 land cover map, which we got from Landsat, compared it to an historical image for that specific area. And we found that there is so much land cover changes in the area. That's conversion of the vegetation into cropland. And so the red images you are seeing there is representing areas that are already being put under agriculture. For entomological surveys, we were using CDC light traps, which were set for four different times over the year, so to cover the wet and dry season. And also for each time when we were sampling, we were setting these traps between 6 p.m. and 6 a.m. for 3 consecutive days. With livestock sampling, as I said earlier on, to collect serum, and at the same time, human sampling to collect serum for screening using ELISA tests. All these data were analyzed using geostatistical models to capture the spatial heterogeneities in terms of exposure. So that's one result of participatory surveys where we have meetings with with local people to discuss the kinds of services and the services they get from these areas. So the main ones which were identified in irrigated areas were, of course, water. This is a very dry area, and with the introduction of the scheme, you would expect that people are now able to get water within their presence or within their settlement areas. And that's one of the questions which we were asking ourselves: why is it during the fallow period, when irrigation is not on, why you still see irrigation allowing water to stand? We soon learned that actually that was designed to help people to go and fetch that water, even when farming is actually not ongoing. For the services, we used proportional piling, which is a common method we used in participatory sciences to capture data on key things that you are asking for. And for this case, we were ranking diseases. The key ones which came out, of course, is malaria and bilharzia. But of course, you know, many people think any feverish illness is malaria. And so there are issues about classifying malaria and other potential infections. That's a result from our entomology, which we comparing periods when irrigation is ongoing, that's when farming is ongoing, and when there's fallow period or when there's resting, when there's no active irrigation. And the key difference I want to show is the brown bar shows areas around the scheme. Those are the pastoral areas not far from the scheme. So it shows that when irrigation is ongoing, you get many mosquitoes being blown or flying to the contiguous sites. But during the fallow period, mosquitoes are confined within the scheme. So you can see that the mosquitoes are not just being during the irrigation period, these vectors go outside the farmlands. The next is a regression analysis of the same result. And the one I want to bring to your attention is the effect of irrigation. It has a much higher impact on the mean densities of these vectors, shown in red. And that's our analysis on human seroprevalence. I haven't given you one for livestock for one sole reason, that the area does routine vaccination for livestock. And up to now, we don't have a test that can differentiate antibodies for livestock, antibodies from active infection from those of vaccination. So for humans, because we really know there's no vaccination for human, this would be a good indicator for infection. And of course, what I'm showing here is there's a higher seroprevalence in irrigated areas compared to pastoral areas, although that's not significant. And I'll be touching on that in a short while. So for discussion, what we are saying is irrigation, as we know it, increases food production. That's one service, but of course there are the services associated with that, which includes land fragmentation and also exposure to disease. And in this area, irrigation is not just for food production. What we realized recently is even pastoralists are now growing pasture under irrigation. And so there'll be more area will be put under irrigation with time. And the catch is these are areas which are conducive for vector-borne diseases, including Rift Valley fever. And so that's why I'm saying the primary vectors for Rift, these areas could actually become reservoirs for the disease in future if no measures are taken to control the vectors at the same time or when you're introducing irrigation. So the last point I want to make is the seroprevalence data that we have now doesn't really show a significant effect. We think it's because these indicate cumulative infection, which have occurred over time. And maybe there's an opportunity for looking for active infections through more intensive surveillance. And so one of the methods which we are suggesting for control is using good technologies for irrigation, for example, using a piped irrigation system, which actually saves water by reducing evaporation, but also controls vector-borne infections, because you really don't have floods, which we have now for free. So yeah, I want to thank the team in IDS and also ESPA for funding.